There has been reasonable rising concern about the Zika virus over the last year and a half. The Aedes aegypti and Aedes albopictus mosquitoes are the primary vector for the spread of this virus. It also rarely shows symptoms, and when it does, they generally present as flu-like in nature. Infected individuals are able to easily move throughout the globe and begin outbreaks elsewhere. The virus may also be spread through sexual activity. Right now, we don't really know how long that persists, and you don't have to show symptoms to transfer the virus. Healthy adults really aren't likely to experience much from having this virus in their system. However, when it comes to a pregnant individual, the virus can attack the growing fetus and cause a variety of health problems later on. A British biotechnology firm known as Oxitec has been testing ways to control the Aedes aegypti population in Brazil for several years. In fact, many internet conspiracy theorists believe that these mosquitoes are the cause behind the Zika outbreak, even though that is not the case. The Oxitec mosquitoes that are released are 99.9% males. They are designed to mate. They do not bite. The main purpose behind these release experiments were to find other methods to control mosquito-borne illnesses. The hope is that with lower mosquito populations, we can virtually eliminate illnesses like dengue fever. To achieve this result, this particular company has taken male Aedes aegypti mosquitoes and modified them by adding 2 different genes. One modification was used to make the mosquitoes fluoresce under the correct conditions so that scientists would be better able to track them. The second gene they nicknamed TTAV, or tetracycline controlled transactivator. For the latter expression, they took a coding sequence from E. coli and the herpes simplex virus and fused them together into one genetic sequence which was inserted into the mosquitoes so that they would produce the TTAV synthetic protein. The production of this particular protein is controlled by TET oocytes, or tetracycline operators. When these mosquitoes are given an antidote of the antibiotic tetracycline mixed with water, the drug interacts with the TTAV protein and binds them together. In the absence of the antidote, the protein binds to the receptor sites, which causes more protein production. These proteins inhibit necessary gene expression for the continuation of the mosquito's life. So in other words, without the tetracycline water, the mosquitoes are as good as dead. There are some experts that are concerned over the release of these mosquitoes, as it has not yet been confirmed whether they are a useful tool or just a money sink. However, currently, Oxitec is being hindered from their FDA-approved release in Florida because of other concerns. It should also be noted that Florida has 80 species of mosquito, and the Aegypti species is invasive. So if we did happen to kill all of them, they would not be missed. If any species do happen to consume the Oxitec mosquitoes, there will be no negative side effects. They are non-toxic, non-allergic, and the genetics of the mosquito will be broken down in the acidic environment of the stomach. The mosquitoes that are being released in these tests have also all been sterilized with radiation, meaning they cannot pass on their genetics. And even if they could, there's not enough tetracycline in the water for their offspring to survive long enough to bite a human being. There is some precedence for success in these experiments, as we have been able to reduce screwworm populations in the United States to pretty much nil through the use of genetically modified organisms. With CRISPR pretty much revolutionizing the genetic engineering field right now, it's not likely that we are going to abandon projects that have potential benefits to our species. If you do have any questions, let me know. I would be glad to hear from you. And please stay safe, my witchlings. Know that you are incredible. And until next time, bye.